Welcome to the next lecture of this course Environmental Biotechnology, and this is lecture number 40, and in this particular lecture we are going to discuss further on the topic of biodegradation of organic pollutants. So, in this particular lecture we would be highlighting some of the very important aspects, particularly the requirements of biodegradation of organic pollutants. Now, fundamental prerequisites for microbial degradation of organic compounds, organic pollutants are very well studied for the last more than maybe 100 years now, and particularly as we understand with the advent of different culturing techniques and characterization of microbial metabolic properties, their genomes, etc. We have gained the considerable understanding about what are the factors, what are the requirements that actually controls the microbial biodegradation of organic pollutants, particularly when it is come to the environmental application of those organisms towards the degradation of the pollutants. Now, one of the fundamental prerequisites for this process is the understanding that biodegradation of pollutants, environmental pollutants, is a self purification process by nature in a polluted medium. So, any kind of polluted environment will have this self purification process set in by virtue of the microorganisms, bacteria, archaea, fungi. They will be naturally growing in that or those environments, and these could be the wastewater treatment plant, contaminated soil or groundwater contaminated with different kind of pollutant like organic pollutants like a leaking underground storage tank, which have been actually a very common phenomena for many of the sites which have been successfully bioremediated. So, this concept of self purification explains that the microorganisms which are there in the natural environment, that is the indigenous organisms present even in the contaminated sites, they will gradually acclimatize with the contaminants present there and will try to grow and will grow definitely, and they will carry out the biodegradation process. However, this self-assembling group of organisms which are having the relevant biocatalytic activities towards the degradation of the organic pollutants, these particular processes require certain conducive conditions, and particularly the growth of specific degraders and the functioning of their enzymes have been identified to be one of the most important criteria for this self purification process. Now, we have already learned two terminologies. One is the engineered bioremediation and another is the natural attenuation. So, if we just try to quickly recap the concept of natural attenuation, where the organisms who are there in the contaminated site itself are allowed to work on the pollutant molecules and they lead to the degradation and the remediation to be achieved. Whereas, in case of engineered bioremediation, we have learned that there could be biostimulation through adding different nutrients or electron donor, electron acceptors, etc., which actually facilitates the existing microbes to degrade the pollutants. Or we can add selected efficient biodegrading organisms, that is called the bioaugmentation. Now, one point, whether we are adopting this natural attenuation or we are adopting a biostimulation or bioaugmentation based engineered approaches, one thing is required, and that is that the presence of the specific degrading organisms. Now, these organisms could be of versatile type. So, there is no boundary that these organisms must be of Pseudomonas type or Bacillus type, may not be. And in many cases, what we have seen that the self purification processes are carried out by a diverse type of organisms. Some of the organisms are individually capable of degrading the pollutants, while some other organisms, they rely on syntrophic interactions between them, and then that syntrophic metabolism favors the degradation of the compounds, particularly when we expect mineralization to be happening. Now, the one part is the growth or the specific degrading microorganisms and their growth. So, their growth should be promoted, and the functioning of their enzymes. So, these are the two things. So, one is the organisms who are to be present there, and the other is the functioning of their enzymes. Okay. Now, if we consider this simple cartoon, that these are the organisms who are capable of degrading or carrying out the complete degradation or biomineralization of organic pollutants to carbon dioxide and water. Now, so one point is that there should be the degrader organism. Let us say some organisms, so these are the degrader organism. Now, the degrader organisms must be capable of having the complete expression of or the production of the required enzymes which are responsible for carrying out the degradation process. So, ideally it is not merely the presence or absence of the organism only, but also it is the presence of the functional enzymes. So, ideally if we consider that this is just, we imagine that this is a microbial cell, and this particular microbial cell, let us assume, is capable of degrading the aromatic compounds like a benzene compound or phenol or toluene or xylene or some other polyaromatic compounds. So, this organic pollutant will be firstly will be taken up inside the cell, and the cells will try to act on that. So, these are the 2 points. Point number 1 is the presence of the organism. So, presence of the bacterium, let us say in this case it is a particular species bacterium. So, bacterium means only one type of bacteria, bacterial strain, one particular type of bacterium, or it could be multiple bacteria also. Now, one is the presence of the bacterium who is identified to be an efficient and is capable of what? Capable of degrading the particular contaminant, in this case this aromatic compound, number 1. Now, number 2 is this degradation process. If we see, we will be able to see, if I just identify this compound as A, that A will be taken up inside the cell, then A will be gradually converted, and it will be converted to a number of intermediates, and then something will be formed. This G will be a part of central carbon metabolism. So, ideally this compound A transported inside the cell, and then A will be subjected to a number of enzymatic steps, and these enzymatic steps will finally lead to the synthesis of some product. That product will be a part of the precursor for the central carbon metabolism, maybe pyruvic acid or acetyl CoA or acetate or something like that. Now, as we understand that presence of these bacteria will be necessary, because this bacteria is having all the enzymes who are capable of doing this process. Now, having said so, it is also necessary to understand that in a contaminated environment, the mere presence is not sufficient. The organism who is capable, who is efficient, who is having all these enzymes, all the enzymes must be expressed, must be produced, and they must be able to carry out the reaction. Now, one part is the expression of these genes responsible for all these enzymes. The other part is that even the enzymes are formed, the microbes might be requiring some other factors for the functioning of the enzymes. As you know, that the functioning of the enzyme is a big aspect, because enzymes are sensitive to different other factors, including the pH, temperature, heavy metals, other organic compounds, and many other things. So, what I said, that the presence of organism is one aspect. It could be very specific degrader like a benzene degrading bacterium or a toluene degrading species or a phenol degrading bacterial strain, something like that, as well as the functioning of the relevant enzyme. the functioning needs their expression of the gene. So these will have basically the expression of the relevant gene expression. So expression of the relevant genes are expression of the relevant genes, number 1, and from these the proteins will be formed, and these proteins will be allowed to function, and this is actually subject to many other factors, whether appropriate conditions are there. So where these enzymes proteins, so these are the enzymes proteins, so whether these enzyme proteins will be able to function or not. So that has to be understood very clearly. Okay, so with that we proceed to the next major aspect of the requirements for this biodegradation. So first is the existence of the organism with required degradation potential. So in the previous slide itself I have emphasized that the organism, that is the capable organisms, are to be there. Now microbial ecology experiments or protocols or techniques guide us to answer this question, that whether the organisms who are capable of performing a particular degradation is there or not, and their relative abundance, whether the organisms are metabolically active, etc., etc. So we know that how to achieve this goal, that the existence of the organism. So this is for example is done, confirmed that the existence of the organisms are there. Now with respect to that, there are certain other points. Points are like these, that the organic compounds will be degraded to a measurable extent. Measurable extent means with which we will be happy. Our environmental people will be saying that yes, this X concentration, X ppm or X gram per kg or gram per liter concentration of a particular substance is now degraded to some microgram or less than microgram per kg or per liter, and within a definite period of time. So that is very important. So they are to be degraded to a measurable extent, and that measurable extent of degradation is going to be achieved only if the organisms has enzymes that catalyze its conversion to a product that can be funneled into the existing metabolic pathway. So it is subjected to the expression of the enzyme that I have mentioned in my previous slide. So the compound has to be converted to some kind of product which can be, like in the previous slide I have drawn, that the product G or the metabolic outcome of all the reactions of the benzene degradation or the phenol degradation, like pyruvate or acetate, will be funneled into the existing metabolic pathways. Now for example, the presence of the organisms is required, and also the organisms will be capable of producing all the enzymes required for converting the product into the target compound to the product molecule, and the product molecule will be fed into the existing metabolic pathway. Now in that respect, I will also mention, before I explain this particular topic or figure, that the metabolism has actually two steps. In one step, for example, this is the organic pollutant, and here we have the cell. So this compound is taken up inside the cell, which I have indicated as A. So A is taken inside the cell, and then there are steps. There are many steps through which the A is converted gradually one by one to some intermediate. Let us say the intermediate name is pyruvate. So in most of the biodegradation processes, we can see that no matter what is the target compound, whether it is a polyaromatic compound, whether it is a monoaromatic compound, whether it is a simple benzene molecule or a phenol or a toluene, or it is a alkane molecule of some carbon chain or more than 10 carbon chain or more than 20 carbon chain. So all these different organic carbons are generally funneled. We called funneling because we can have actually here diverse types of organic compounds. They will be using maybe different steps, but eventually they will converge on some reaction, some products, these products will be something like this pyruvate. Now, what is the significance of, for example, pyruvate? It may be acetate also in some cases. So, pyruvate or acetate. Now, what is the significance of having acetate or pyruvate? Biodegradation of the contaminant, organic pollution goes on, as I said, that goes on in two steps. So, this is the first step, step A, where the pollutant will be converted to some molecule which is a part of, I will write that, central metabolism. So, what is central metabolism? In any microbial metabolic process or metabolic landscape, we see that there are certain central metabolic processes are going on. For central carbon metabolism, central nitrogen metabolism, central sulfur metabolism. So, these are the bunch of pathways, bunch of steps, bunch of reactions who are facilitating conversion of different substrate to different products and intermediates, and these intermediates are feeding into the bulk precursor pool, so that everybody or every other reactions running in the cell can utilize those metabolites. So, this is the purpose of the central metabolism, and they are often generating lots of energy and reducing power, particularly when it comes to the central carbon metabolism. Now, these acetate and pyruvate, which are produced as part of the phase A of the biodegradation, are actually very important intermediate or starting molecule for the phase B, which is the central metabolism. For example, you possibly all know about the citric acid cycle or the TCA, tricarboxylic acid cycle, where the pyruvic acid or the acetate molecules can be completely metabolized to carbon dioxide and producing lots of energy, as well as other intermediates like succinic acid, alpha ketoglutarate acid, and all these alpha ketoglutarate etc. can be utilized in different other cellular biosynthetic reactions. So, the central metabolism is allowing the complete oxidation of the substrates. So, both A and B are required. If we leave it only on A, then suddenly the B part, because central carbon metabolism is anyway growing in the cell, all cells they are performing central carbon metabolism. So, eventually it will be running either through TCA cycle or it will go through fermentation process. So, one of the fundamental requirement will be that the organism is capable of degrading this through the phase A, as I mentioned, to a measurable extent, so that the organisms will be able to feed these products of this phase A into the central carbon metabolism. Central carbon metabolism is already existing in all cells. All cells, all microbial cells, bacteria, archaea, they perform the central carbon metabolism. So, biodegradation will only provide the substrate which will be the starting molecule or some intermediates of the central carbon metabolism, so that they will continue. Now, and as we can see in the panel, the figure that is presented here, now these phase A type of reactions could be very dedicated reactions, like I will discuss about these dedicated reactions, maybe I will show you some examples, and they will produce products like hydrogen acetate etc. Now, some of the microbial strains, they themselves are capable of utilizing them, as I mentioned. So, they will be straight away going to the central carbon metabolism. So, maybe this particular bacteria, they will themselves use this acetate for their own central carbon metabolism, and they will generate energy out of it. But it may so happen that they release part of this acetate outside, and other organisms will be enjoying this acetate or pyruvate as a substrate for their carbon and energy source, and they will grow happily on them. So, they could be nitrate reducers, they could be iron reducers, they could be sulfate reducers, and they will produce more biomass and some other metabolic products also. And this is particularly true when we have an anaerobic condition. If it is not an anaerobic condition, then possibly some aerobic bacteria. So, some aerobic metabolism by some aerobes will be performing that. Otherwise, this organism will be itself may be capable of degrading this acetate or pyruvate completely. So, one single bacterium or bacterial strain can be performing the complete degradation, or it could be a kind of a joint venture where different other organisms are also associated with the degradation process, particularly when we see the process of syntrophism, because in the hydrogen and acetate both are utilized by different methanogenic organisms to facilitate the degradation of the organic pollutants. Now, the second point with respect to this is that it is not only that the candidate organism would be there and the candidate organism will be having just the biodegradation steps. It is not just a biodegradation step or a biodegradation reaction. It is actually a multi-enzymatic process. So, in most of the cases we see that you might have noticed that earlier I have drawn A prime to B, B to C, C to D, etc., etc. Similarly, if we look at the biodegradation of most of the compounds, these are truly multi-step and many-step reactions. So, in each of these steps, we will have involvement of specific enzymes, and these specific enzymes are going to play a role in catalyzing the or the converting the one substrate to an intermediate, and the intermediate will be converted to another one. So, let me show you something which is called the benzene or benzoate degradation pathway. So, this pathway is obtained from the KEGG database, and the link is provided here. So, you can always use this link to study more. And now, I will highlight the benzene degradation part. So, here the entire pathway is for benzoate degradation, and there are many reactions involved in benzene reaction. One thing I will show you that we will take a part of this and then we will. So, here you can see that benzoate is written, and this is actually degraded through phenol and finally through catechol, because catechol is one of the major intermediates of this degradation process. It happens inside the cell. So, the benzene is taken inside the cell and it is metabolized. So, you can see there are multiple steps. So, all these numerical values, these are actually the EC numbers, enzyme classification numbers, and in a live form, in an online form, if we just click on that, it will show us all the details of these enzymes. So, you can see that there are multiple possibilities through which the benzene or benzoate can be converted to, for instance, the catechol, which is found to be a very important metabolite. I will discuss little bit more on this catechol. Now, I have enlarged this particular part which we wanted to actually discuss in a little more detail. So, this is the one of the sections of the benzoate degradation, where you can see that the benzoate is converted to catechol, and this catechol is subsequently catalyzed through multiple reactions, and then all these multiple reactions, I will enlarge it further. So, all these multiple reactions eventually led to what? Led to, if we consider catechol, so one line is highlighted here, I have marked it for your understanding, that you can see that the catechol is converted to some semialdehyde first, to hydroxy muconate semialdehyde, because this is one of the pathways. There are many pathways which can, so you can see, for example, there is one pathway running over here, there is another pathway which may go like this and that, this side. So, there are multiple pathways. So, we are talking about a red colored pathway. So, you can identify that the catechol is converted to semialdehyde, muconate semialdehyde, and then muconate semialdehyde can undergo two sets of reactions, either in this way or in this way. But essentially, whether it goes, it will be converted to that oxopent 4 enol and then hydroxy oxopent phenoate, and then it will be forming the acetaldehyde and pyruvic acid. And this acetaldehyde and pyruvic acid will be directly fed into the citric acid cycle or TCA cycle, because that is a very known pathway of the central metabolism. So, these acetaldehyde and these pyruvate molecules will straightway be moving towards the central carbon metabolism, which will facilitate the further oxidation. And it is not only oxidation, along with oxidation they will produce the many intermediates. Those intermediates will be again acting as a precursor. Otherwise the complete oxidation will lead to the formation of the energy and the reducing power which the cell will enjoy. So, these are the some of the enzymes I could retrieve. Now, these as I mentioned, that these degradation processes can be happening under both methanogenic condition, where electron acceptors or other electron acceptors are possibly not there. So, directly in the lack of oxygen it is possibly fermented, and following fermentation some of the target compounds will be converted to hydrogen and acetate, and then different kind of methanogenic organisms will facilitate the utilization of hydrogen and acetate, and ultimately they will make the entire process thermodynamically favorable. And it is actually an example of the interspecies metabolite transfer, like hydrogen and acetate, which are produced from the pyruvate and acetate, which are produced. In this case we have already noticed that we have this acetaldehyde and acetate all are being produced. So, if the oxygen is not available or terminal electron acceptors are not available, then this pyruvic acid and this acetaldehyde will straightaway be going towards the fermentation, and following the fermentation they will produce the hydrogen and acetate that will be used by the methanogens. However, during the presence of other electron acceptors, for example, if oxygen is consumed or oxygen is not there in the particular contaminated environment, in most of the cases we see the oxygen used to be there at some concentration level, but eventually the oxygen is used up by the aerobic organisms. So, I will come to that point. So, then the other electron accepting reactions like the nitrate, sulfate, iron, etc., they will facilitate the process of the degradation. Now, with this respect, another and very interesting point is, the greater the differences in chemical structure between the target pollutant and the constituents of the living organisms, the less likelihood that the contaminant will be recognized as a substrate. So, that means the target compound, we have discussed that the benzene is taken up inside the cell and is degraded up to acetyl-CoA or acetate or pyruvate. Now, if the benzene molecule is not identified as a substrate, what will happen? Fortunately, benzene will never be identified not as a substrate, because microorganisms have evolved with this kind of petroleum derived hydrocarbon residues or chemical structures for their long period of coexistence during the course of evolution. But in case some new molecules come, or there is a kind of modification in the existing chemical structure of the alkane or maybe in the aromatic rings, the microorganism, if they are unable to recognize them as substrate, then they will possibly not be able to facilitate the degradation process. This hold for the cases of a pollutant that occurs less commonly, particularly if a pollutant is there which is less commonly present as a building block in the living matter, like the particular pollutant molecule has never been seen by the microorganisms, or part of its products or chemical structures are not there in the central metabolic pathways, etc., then extensive degradation is unlikely to occur, because this molecule seems to be new to the cellular system, not only from a recognition point of view, but also the chemical architecture of the entire cell possibly recognizes this molecule as a foreign molecule and is a new molecule for the cell, because it is not resembling any of the intermediates present in the cellular system. Now, some structural features in organic compounds that are not common in nature are called the xenophores. The xenophores are very common in terms of when you see the recalcitrance of some compounds are discussed. The presence of the xenophores, like the substitution of chlorine, nitrite, cyanide or SO3 groups in a particular organic compound, these are called xenophores, and these substitution of these groups led to a kind of make them kind of unrecognizable for the microbial system. Now, contaminants that contain such xenophores tend to be recalcitrant to microbial degradation, because after these xenophore molecules are present, the microbial systems will feel something that this is a kind of a absolutely new molecule to me, I don't know how to deal with this. Now, in that respect, we should talk briefly about the microbial infallibility hypothesis, which is a very well known hypothesis with respect to biodegradation, and it says that the virtually for any available organic compound there may be an organism that will break it down under suitable conditions. So, all organic compounds present available to the microorganisms will be definitely degraded at some point of time at some suitable conditions. If not, if the compound is not getting degraded, then evolution and adaptation will produce such organisms, such bacteria, archaea or fungi, which will in a relatively short period of time, which will facilitate this degradation process. The second prerequisite is the presence of specific degrader in the contaminated environment. Now, hydrocarbons have a natural pyrolytic origin that I have discussed, and for the evolutionary origin, and have been in contact with microorganisms throughout the eons of geologic time. So, microbes are interacting with hydrocarbons, aromatic and aliphatic. So, for them it is not at all a new molecule. It is not surprising that many bacteria have evolved and acquired the ability to utilize hydrocarbons as a food. So, when we started working on the hydrocarbon degradation, biodegradation, scientists were very surprised initially, but very soon they have realized that as if it is a regular food for them, because there are plenty of natural environments, underground aquifers, etc., where different concentrations of naturally produced hydrocarbons are present, and microbes are regularly utilizing those hydrocarbons as their food in a sense that it is a source of carbon and energy from them. The ability of microbes to degrade, for example, the benzene, toluene, ethyl benzene and xylene, which is referred as the BTX, has been known since 1908. You can imagine that 1908 the Bacillus hexaborum later was identified by virtue of the ability to grow on toluene and xylene aerobically. So, there have been numerous studies and reports, and we have a whole plethora of information and organisms available for that. The next point would be, requirement would be the accessibility of the target pollutant to the microorganisms. The pollutant molecule is there in the environment, and the candidate organism who can degrade the pollutant is also there, and we expect that the pollutant organism is capable of expressing all its genes, everything is fine, is all set to degrade the compound, but the degradation may not proceed with a kind of expected rate or expected extent, because there would be some other limitations, could be limitations. These limitations are considered as the limitation from bioavailability point of view, and because the lack of adequate contact between the pollutant and the microorganisms. The target pollutant must be accessible to the bacterial strains or the archaeal strains or the fungal strains, and unless they are in the aqueous phase, because microorganisms will work only in the aqueous phase. So, the pollutant molecule should be available in the aqueous phase, from where the microbes will be able to interact, and therefore, the rate of biodegradation can be controlled by the diffusion, desorption or dissolution rates, which are actually applicable for the contaminant molecule, the organic molecule which is present along with the soil particles or the rocks or the micropores or the nanopores inside the rock or crust or within the aquifer sediments. So, how quickly or what extent these hydrocarbon molecules are getting diffused out, so that the microbes are able to interact with them, or they are dissolved out, or they are dissolute out. Now, microorganisms generally assimilate the pollutant from the liquid phase only, and cannot effectively degrade a pollutant until it dissolves from the aquifer solids, etc. So, one picture that I will show you here, this is published in this paper, and you can access and go through this paper with respect to all the details. So, if you consider a mineral fraction means it is a rock or some minerals present in the rock. You can see that there are many small habitats kind of things are there, habitat for the zones where the organic pollutant can have their specific interaction with the mineral fraction, and they remain in that, like a non-aqueous phase liquid fraction, the bacteria will not be, so if we consider the bacteria will not be able to interact or degrade it unless this non-aqueous phase liquid fraction is dissolving out and then available in the aqueous phase liquid. Surface option to diffusion into pores. So, in the mineral phase there is pores in which the organic pollutants are residing and the microbes are unable to react with them. The spherical structure is for the microbe. So, microbes are unable to reach out there because they are sitting inside and they have a stronger interaction with the mineral fraction. So, ideally we need to consider the fact that how actually the pollutant organic molecule will be available to the bacterial strains or the microbial strains. So, one aspect could be that the production or the availability of different biosurfactants by the microbes. So, microbes often we have found that they produce these biosurfactant molecules to emulsify or solubilize or make these hydrocarbon molecules available to them. So, other factors which control the bioavailability could be the bonds requiring the cleavage. Cleavage means where exactly the enzymes are going to attack on the molecule, the organic molecule structure must be exposed and not be blocked by large atoms such as a chlorine molecule or so that each of the major bonds which are supposedly the target region for the enzymatic catalytic reaction, they should not be kind of under some kind of steric hindrance. And other one is that the target molecule or the target pollutant molecule must be able to pass through the cellular membrane quite easily. Now, many common priority pollutants like the BTX, benzene toluene ethyl benzene xylene or trichloroethane kind of compound are relatively soluble and bioavailability rarely limits their biodegradation. So, they happily move into the cellular system and cells are able to degrade with them very quickly. However, the polyaromatic hydrocarbons and PCBs, for example, they are more hydrophobic and therefore the microorganisms they face different levels of challenges in terms of interacting with those. The next one is the induction of appropriate degradative enzymes. So, as we have seen already that the microbes need to actually deploy a set of enzymes to facilitate the degradation process. Some of the enzymes, particularly the central carbon metabolism part of the enzymes, are constitutive enzymes. Most of the microbes, as I mentioned, they are expressing those enzymes. But for the specific degradation parts, microorganisms need to induce some of the genes, some of the enzymes, and the presence of the inducer and the repressors are very relevant, because inducible enzymes are induced only when the inducer molecules are maybe a lower concentration of the organic pollutant itself would actually facilitate the induction of those genes into from their genome. On the other hand, the presence of some readily metabolizable other substrates would repress the expression, like presence of ethanol. For example, if you have ethanol along with these organic pollutants, you will see the ethanol is getting metabolized rather than the organic carbons, because the genes are not getting expressed because of the some intrinsic repression which are posed by the other repressor molecules. The last point for today's lecture regarding these biodegradation process is the availability of the electron acceptors and or electron donors. This is another very important aspect, because microorganisms, why they are interacting. I said sometimes that it is used, the organic pollutant molecules are biodegraded because they want to use them as food, as the source of their carbon and energy. That means the microbes, they try to obtain the energy from these molecules, these organic pollutant molecules, to drive the life functions, either by producing the ATP or the reducing power or the proton motive force and the precursor molecules. And these reactions are complex set of reactions, very complex set of reaction. I have shown you some part of the KEGG pathway for the benzoate reaction. You have possibly noted that there are several reactions going on. And the microorganism, they express all these genes and produce the enzymes in order to facilitate the oxidation of the target compound wherever oxidation process is applicable for the biodegradation. So, it is basically not only oxidation but also a complex sequence of oxidation and also reduction process. because somewhere the electrons must be transported to a electron to an electron acceptor, because if no electron acceptor is available to accept the electrons, then the entire process will stop. So, it is not a merely oxidation process only, it is actually oxidation in one hand and reduction on the other hand. Other hand in the sense some electron acceptors must be available and must be playing roles in accepting the electron, so that the flow of electron goes on. Now, where the electrons are transferred from electron donor, that is a fuel molecule, to the electron acceptor. So, we have already discussed this part of this thing, that the contaminants can serve as electron donors, most of the cases where oxidation is involved, and wherever the oxidation based biodegradation is happening, all the oxidizable pollutants, they are being utilized as a source of electrons, and the electrons are being transferred to electron acceptor. So, these are the electron acceptor. So, under the aerobic condition, oxygen is the electron acceptor. Under anaerobic condition, different other electron acceptors, they play a role. And so, basically, as the organic pollutants are degraded to oxidized product, let us say the pyruvate or acetate or maybe complete degradation, the role of these electron acceptors are very critical. If electron acceptors are not available, then possibly this process will not proceed. This process will collapse, because where the electrons will go finally, because we already have discussed this part, and I will just quickly try to remind you that the organic pollutants are actually getting oxidized. So, the electrons are coming out. Now, these electrons should be taken up by somebody. So, who is going to take it? It is the, let us say it is the NAD plus who will take the electrons and will turn into NADH plus H plus. Now, these NADH H plus should donate the electron to someone and then turn into NAD plus. So, this reoxidation of NADH H plus must occur, because the cell maintains a definite pool of NAD plus and NADH H plus. If they have more of NADH H plus accumulating in the cell, that means the cell will get a signal that we are energy sufficient, we have more NADH H plus, that means they are carrying electrons. So, we do not need to carry out any further oxidation. So, eventually this may be stopped. So, they need to reoxidize in order to facilitate the organic pollutant. So, when we think of a bioremediation on a field level or a practical process or in a practical setup, in a real contaminated environment, we need to assume or we need to consider that where these electrons will go finally. So, electrons will be obviously going to the terminal electron acceptors. So, then if we consider that NADH plus H plus is having the electrons and wants to come back to NAD plus, wants to convert to NAD plus, that means these electrons must be given to someone. So, who is this someone? So, these are the electron carriers, as I have already mentioned in one of my classes. So, these electron carriers, they carry the electrons, and finally the electrons are given to the terminal electron acceptor, that is in under aerobic condition or aerobic condition is the oxygen. So, oxygen is getting converted to water. But if oxygen is not there, or if the oxygen is consumed, in most of the contaminated river or lake or other ecosystems, we see that the oxygen is consumed very quickly, because the aerobic bacteria, aerobic microbes, they try to utilize the presence of oxygen as the terminal electron acceptor, and they degrade the organic pollutant, and therefore the oxygen level declines, which is often connected to the event which is called the BOD of the water particularly. So, BOD refers to basically that the biological oxygen demand, it is indicating the organic pollutant load. Organic pollutant load in the sense, because if you have higher amount of organic pollutant, obviously the oxygen will be consumed more. So, the oxygen level will fall quickly. Now, the drop in oxygen level will give you some idea that how much organic pollutant was there. So, basically what I want to mean or discuss over here, that the oxygen is preferred, because under aerobic condition it is the thermodynamically most preferable substrate. The microorganism will be very happy, they will grow utilizing the organic pollutant, and they will grow, but very soon the oxygen level might fall down because of diffusional barrier and many other factors. So, it will switch to the alternate TES, and these alternate TES will now play a very important role. And what we have seen, that the classical thermodynamic concept implies that the following sequence of electron acceptor utilization is mostly predominant in organic pollutant contaminated natural environment, particularly under aquifer conditions or underground condition, where the level of oxygen is highly controlled, because oxygen is only produced most of the cases minimally, and it is actually the diffused oxygen from the top of the soil activities. So the oxygen level declines. Naturally, the aerobic processes will gradually play minor roles, and the denitrification process using nitrate as electron acceptor, manganese reducing bacteria, then manganese reducing process followed by iron, sulfate, and eventually the methanogens will come into play, where they will try to use the carbon present, or the carbonate or carbon dioxide as the electron acceptor, and they will produce methane, and that all this process will drive the biodegradation of the organic compound. But based on the thermodynamic gain that gives free energy which could be produced, the extent of the degradation will also vary. Obviously, this will be the producing the maximum Gibbs free energy, so it will be most preferred, and the extent of organic pollutant degradation is expected or may be expected to be very high. So it will go on, like the extent of degradation will also follow the same trend. So in a sulfate reducing condition, the extent of degradation will be much lower compared to the nitrate reducing condition and compared to the aerobic condition. And under these methanogenic conditions, where no other electron acceptors are there, the biodegradation will be highly restricted. However, in many of the contaminated environment, we see that the sulfate reducers and these methanogens, they play a role together, that is called the syntrophic metabolism, and through which they facilitate somehow the better degradation of organic pollutant up to some extent. And in many of the US DOE sites, for example, this application of this and in marine environment also, what we have noticed that the presence of sulfate, it prefers the, or it favors the biodegradation of organic pollutants, because many pollutant degrading bacteria, they use sulfate as terminal electron acceptor and convert the organic pollutant into acetate or other secondary fermentation products like hydrogen, and those products are utilized by the methanogenic organisms, thereby allowing this interspecies metabolite transfer, and overall that process facilitates the degradation of the organic pollutant. So for this lecture, I advise you to consider these articles. So most of these articles are found to be very useful, particularly the book written by this particular book, that bioremediation and natural attenuation, and also the presentations of some of these articles are found to be very lucid, and one can easily understand the importance of the different aspects of these bioremediation processes. So in conclusion, in this class we have discussed about the major requirements of biodegradation. We also discussed about the role of bacterial metabolic pathways, enzymes, induction of such enzymes, bioavailability of organic pollutants, etc. Importance of the availability of appropriate electron acceptors are also discussed. And thank you so much.